Hello, everyone. My name is Neeraj Tiwari, and I'm a technical information scientist at the Jackson Laboratory. I'm delighted to share this educational information to support you in achieving your drug discovery goals. And today we are going to talk about several key features of using a preclinical assessment of investigational gene therapy products for neuroscience. And that is in the context of rare neurodevelopmental and neurodegenerative diseases. So we'll start today's webinar by introducing gene therapy, different type of gene therapy tools available, and then discuss some challenges this field is facing. Then we'll go over examples of gene therapy preclinical studies done here at JAX by our in vivo pharmacology services group to correct rare neurodevelopmental and neurodegenerative diseases. And finally, we will feature mouse models and study designs our in vivo pharmacology services uses routinely that can be used for preclinical evaluation of gene therapy. So let's get started by introducing gene therapy and different types of gene therapy tools available and the challenges this field is facing. So as you are aware, gene mutations are either inherited or may occur throughout our life. These mutations can affect the way our cells and body function. Gene therapy can modify disease-causing mutations to stop or slow the effects. And according to US Food and Drug Administration, gene therapy is a technique that modifies a person's genes to treat or cure diseases. Gene therapy can work by several mechanisms, according to FDA, by replacing a disease-causing gene with a healthy copy of the gene, inactivate a disease-causing gene that is not functioning properly, and introducing a new or modified gene into the body to help treat a disease. To give you a historical perspective on the development of gene therapy, let's take a look at the figure on the right side. So in the early 1960s, for the first time, scientists demonstrated that a genetic defect could be rescued by transferring functional DNA from another genetic source. In the 1980s, AAV2 genome was cloned and sequenced, and the AAV vector was used for in vitro gene delivery. Development of RNA and DNA editing tools since the beginning of new century is going to further advance the growth of new gene therapies. Let's take a look at progress of gene therapy use in clinical trials for rare diseases. So in regards to gene therapy clinical trials for rare diseases, I want to share this graph that provide insight into where the field is at and how it's growing. There has been a consistent increase in the number of gene therapy clinical trials in rare diseases since 2012. Next, we will find out the delivery approaches used for gene therapy. So there are two approaches, which includes in vivo strategies and ex vivo strategies that is used for delivering gene therapy. And the ex vivo strategy involves collecting cells from the patient, modifying the DNA, and then putting them back into the patient. In vivo gene therapy is when gene is delivered directly into the patient to make the modifications in the targeted cells. Because in vivo method is currently the preferred way of treating nervous system diseases, for today's discussion, our main focus is is going to be in vivo gene therapy. And some of the main tools that we are about to discuss includes antisense oligonucleotides, in short ASOs, gene editing tools, viral vectors, and non-viral vectors. Next, we'll briefly discuss each of these gene therapy tools, starting with ASOs. So ASOs are RNA-based editing tools that changes the gene expression at the transcript level. They do not make any permanent changes in the genome. ASOs are artificial nucleic acid sequences that bind to RNA and influence the maturation of the pre-RNA to mature mRNAs and protein. ASOs can be used to select mRNAs for RNase H-mediated degradation. They can also be designed against primary transcript to cause alternate splicing. Because ASOs are single nucleotide sequences, they are degraded easily. Modifications have been made to increase ASO's stability, protect against nuclease degradation, improve cellular uptake, recruit RNase H, and reduce immunogenicity. Next, we'll show use of gene editing tools as gene therapy. So genome editing technologies allow genetic material to be added, removed, or altered at exact locations in the genome. CRISPR-Cas9 is a well-known genome editing tool. CRISPR-Cas9 can be used for canonical and non-canonical DNA editing. As shown in the figure on the left, the canonical DNA editing directed by guide RNAs directed by guide RNAs, Cas9 nuclease induce double-stranded breaks. These double-stranded breaks are then repaired by non-homologous end joining or by homology-directed repair. And what happens with the non-homologous end joining? We can either knock out the DNA or knock out the sequence, undesired mutated sequence, which the SDR we can actually introduce by using donor templates. We can insert desired sequences of point mutations in the host genome. For non-canonical DNA editing, Cas9 nuclease doesn't create double-stranded breaks. For this, a catalytically dead Cas9 or nickase Cas9, which is also known as nCas9, is used. These Cas9s are fused with other proteins to alter single nucleotide base pairs, which is also known as base substitution or base editing. And they can also be used for making alterations in DNA domains using prime editing to make the gene repaired. So next we'll discuss vector use for gene therapy delivery. So the therapeutic genetic material for gene therapy can be delivered into a cell using viral vectors and non-viral vectors. In order to make viral vectors safe for use, all the viral genes are removed and the vector is modified to deliver only therapeutic genes. There are 4 main types of viral vectors used for gene therapy, which includes adeno-associated viruses, in short AAVs, adenoviral vectors, lentiviral vectors, and retroviral vectors. For AAV and adenoviruses, their genetic material is not integrated into the chromosome in the human cell. For lentiviruses and retroviruses, their genetic material integrates into the chromosome. The image on the left is showing gene therapy delivery of AAV into the cell. And so the AAV is bound to the receptor, which is endocytose in the lysosomes. This AAV is then delivered to the nucleus where the genetic material is delivered into the nucleus. And this single-stranded AAV genome is then converted into double-stranded DNA. This is then translated and transcribed and translated to produce the therapeutic protein. Next, we'll discuss non-viral vectors for delivering gene therapy. So a non-viral vector is a delivery mechanism that doesn't use a virus for the delivery of genetic material. Non-viral vectors are designed using natural and synthetic materials that are compatible with the human body. These include lipids, polymers, and nanoparticles. Lipofectamine and gold nanoparticles have been used to deliver nuclease, mRNA, proteins, and guide RNAs. So lipid nanoparticles are currently used for as non-viral vehicles for genome editing cargo delivery in vivo. These can be developed for treating nervous system disorders. Next, we are going to talk about some challenges with gene therapy strategies. Gene therapy has its challenges. Efficacy is the number one, such as in case of ASOs, repeated administration is necessary for persistent RNA correction. Safety concerns, such as inflammatory reactions against viral capsids used for viral vector-based therapy, and bacterial nucleases used for gene editing tools. Delivery of proteins and nucleic acids to target cells and tissues are affected because of neutralizing antibodies. So pre-existing neutralizing antibodies may reduce efficacy in case of viral vector therapy. Blood-brain barrier is a limiting step for most of the CNS treatment. And in order to circumvent this problem, various specialized routes of delivery to the nervous system has been developed. In the next slide, we'll discuss routes of delivery to the nervous system. So figure on the left shows in vivo routes of gene therapy to the nervous system in humans. In case of AAV, intraparenchymal infusion remains the most commonly used approach to deliver a therapeutic gene in the brain. For multifocal CNS disorders, we use CSF delivery via intracerebroventricular, intracisternal, and intrathecal injections, and also via bloodstream. For motor neuron diseases, that can be used to improve. So for motor neuron diseases can be improved by intramuscular and intraspinal AAV injections. So local injections to the vector, local injections of vector to the eye or to the cochlea are chosen for the treatment of neurosensory disorders, as a relatively small area is required to be targeted. An alternative and non-invasive option can be intranasal delivery that can be used for secreted proteins that can diffuse through the CNS. We require similar delivery routes for preclinical testing of gene therapy tools in mouse models of nervous system diseases. The table shown on the right side are dosing routes available for mice at JAX for preclinical in vivo services. In the next slide, I will talk about the phenotypes that can be monitored in the mice at JAX for preclinical studies related to nervous system disease conditions. So at JAX, we have experience testing gene therapy compounds that may revert specific phenotypes in preclinical models. Phenotypes we have experience working with includes survival phenotypes, seizures seen in many neurodevelopmental diseases, motor coordination and electrophysiology for neuromuscular diseases, behavior seen in many neurodegenerative diseases, and molecular biology and biomarkers. In the next few slides, while discussing case studies, we will see examples of phenotypes measured. So in this session, we'll go over examples of gene editing studies applied to correct rare neurodevelopmental and neurodegenerative diseases. This body of work is done at JAX by our in vivo pharmacology services group. At JAX, we have experience running over 250 studies in the gene therapy space. We'll start with gene therapy treatment using Charcot-Marie-Tooth type 4J, which is in short called CMT4J mouse model. So Charcot-Marie-Tooth disease, CMT, is a group of inherited diseases, disease condition caused by mutations in over 80 genes. And CMT type 4J is a rare disease caused by recessive mutation in FIG4 gene. It leads to neurological conditions that are generally severe and currently untreatable. The FIG4 gene codes for a phosphatase that exists in a complex with other proteins and is involved in phosphatidylinositol 3,5-diphosphate formation. Phosphatidylinositol 3,5-diphosphate has been implicated in the autophagy pathway and lysosomal function. FIG4 mutant cells exhibit extensive vacuolation and have impaired lysosomal function. FIC4 also interacts with TRPML1 channel and dynamin-1 that is again involved in lysosomal fission. And as we can see on the image on the right side, this shows cultured embryonic fibroblasts from FIC4 null mice with accumulated large acidic vacuoles, which ultimately cause cell death. So for the study, the FIC4 deficient mouse, also known as pale tremor mouse, have a very similar phenotype like humans, with demyelination, loss of motor neurons, and vacuolization in many regions of the nervous system. This mice has a median lifespan of approximately 35 days and presents with other phenotypes such as small body size, tremors, and hydrocephalus. Because both humans and FIC4 deficient mouse develop CMT4J phenotypes due to mutation in FIC4 gene, we use this mice as a proof of concept preclinical model for this gene therapy study. In this study, we have used adeno-associated virus serotype 9 vector to deliver wild-type human FIC4 gene to neuronal and non-neuronal cells in a mouse model of CMT4J. So the top image shows the vector is a single-stranded AAV9 carrying the wild-type human FIC4 cDNA, driven by a ubiquitously expressed chicken beta-actin promoter with a CMV enhancer. In the bottom image shows the preclinical study design, where the mice were injected at different time points based on pre and post disease onset. Also, different routes of administration was also tested. In-life readouts such as survival, body weight, electrophysiology, and behavior was conducted. Also, post-mortem analysis, post-life readouts such as FIC4 protein levels, histopathology, AV9 FIC4 biodistribution were measured. So this data shows JAK's capabilities to deliver gene therapy with multiple routes at multiple ages and time points. And we can measure survival in mice following delivery of AAV9 vector. So pre-symptomatic intervention was done in FIC4 null mice in which AAV FIC4 was delivered through ICV. ICV at P1, postnatal day 1 and postnatal day 4 stages. This image shows that almost 90% of the mice that were injected at postnatal day 1 showed survival for more than 12 months of age. Also, the mice that were injected at P4, postnatal day 4 stage, showed almost 65% of mice survived more than 12 months of age. The control mice that were injected with FIC4 AAV, almost all of these mice survived. 100% of these mice survived, which shows the safety of the viral vector. So, since post-onset treatment and intrathecal delivery is more translationally relevant for clinical trials, we did post-symptomatic interventions in FIC4 null mice, in which FIC4 was delivered through intrathecal injection at postnatal day 7 and postnatal day 11 stage. P7 injection resulted in increased lifespan in these mice. So the mice survived at the age like almost 90 days. The injections that were done at P11 stage, the mice didn't show increased lifespan as compared to the FIC4 null mice. All these group of mice, they showed some growth and gained weight compared to the FIC4 null mice, but the weight they gained was not close. It was always less than the control mice. So these results indicate that the treatment with AAV9 FIC4 is able to improve survival, growth of FIC4 null growth in FIC4 null mice, and the success of the treatment was dependent on the age of injection. This data shows JAK's capabilities to determine tissue biodistribution of AAV-delivered gene therapies. For the mice treated with AAV9-FIC4 tissue, AAV9-FIC4 tissues were collected at 6 months of age after ICV treatment and 4 months of age after intrathecal treatment. The vector copy number was assessed by qPCR. With the gene therapy, we want to be target tissue specific. So as we can see that the AAV FIC4 expression was higher in brain and spinal cord, where we actually want the expression to be, and it was clearly very showing clearly very less expression in kidney, ovary, and testis, where we don't want the expression to be. So this just shows that we can do biodistribution studies here at JAKS. So this data shows JAK's capabilities to evaluate effectiveness of AAV-delivered therapy using molecular biology. So these experiments assessed effectiveness of AAV FIC4 to induce human FIC4 expression. We used Western blot to look at FIC4 protein levels in whole brain and spinal cord. So the mice that were, the Western blot shows that the mice that were that lacked FIC4, we don't see any expression of FIC4 proteins in these mice. The mice that were treated with AAV FIC4 at postnatal day 1 and 4, they showed increase or similar level of FIC4 protein expression as compared to the control mice. Also, the mice that were treated with AAV FIC4 at postnatal day 7, whether with the undiluted and diluted vector, they showed decreased expression of FIC4 protein as compared to the control mice. So this data shows JAK's capability to quantify CNS pathology in AAV-delivered gene therapies. Histopathological analysis was performed in ICV and intrathecal AAV9 FIC4 treated and untreated FIC4 null mice and wild-type controls. Untreated FIC4 null mice showed pronounced vascularization in dorsal root ganglion, spinal cord, and cerebral cortex at 23 days of age. In contrast, mice that were treated with ICV injections, after 6 months of injections, the degree of vascularization was completely rescued in the dorsal root ganglion, spinal cord, and the cortex. The mice that were treated intrathecal injections at postnatal day 7, they showed some rescue similar to ICV injections in the dorsal root ganglions and spinal cord, but there were still some vacuoles present in the cortex. So, when we did this similar ICV AAV FIC4 injections in the control mice, we don't see any vascularization in the cortex, spinal cord, and DRG, which shows the safety of the treatment. So next we'll discuss gene therapy treatments used in SMA mouse model. So spinal muscular atrophy, in short, SMA, refers to a group of autosomal recessive neuromuscular disorders. The mutations happen in the survival motor neuron 1 gene that causes a deficiency of survival motor neuron SMN protein. This results in degeneration of neuronal cells in ventral horn of the spinal cord, leading to progressive muscle weakness and atrophy. Severity is correlated to the level of SMN in lower motor neurons. In the most cases, lack of SMN results in paralysis, loss of the ability of to breathe or swallow, which ultimately leads to infant mortality. Due to an evolutionary recent duplication, two SMN genes, SMN1 and SMN2, exist on human chromosome 5q that only differ by 5 non-polymorphic nucleotide changes. One of these differences, a C to T transition located in exon 7 in SMN2, produces a protein that has truncated, which is truncated and is degraded very quickly. So in normal individuals, SMN levels produced by these two genes support the development and maintenance of the motor units. However, in patients with mutations in SMN1, SMN levels produced by SMN2 are not enough to support the development and maintenance of healthy motor units. As shown here, the phenotypes involve neuromuscular dysfunction and denervation. So for these studies, the Bergeron model is a mouse model that is representative of type 2 and 3 SMA patients is used, which develops defective neuromuscular junction maturation at postnatal day 10. Other phenotypes seen in this mouse model includes necrosis in the tail, as well as cardiac abnormalities that impact survivability in these mice. This is particularly useful model for efficacy studies due to its wider window for therapeutic intervention. In this study, antisense oligos administration at postnatal day 10 and 25 rescues lethality, the cardiac phenotype, and tail necrosis. ASO therapy at age of onset dramatically rescued the survival and weight gain of the Bergeron mutants. These mice benefited in response to the treatment, and the earlier the intervention, the better the outcome was seen in these mice. So this data shows electrophysiology phenotype of the Bergeron SMA mouse model and rescue by ASO therapy. In image A, compound muscle action potential tested the collective output of the muscle fibers due to nerve stimulation. CMAP values improved in the group treated with ASO at postnatal day 25 compared to the vehicle-treated group. In image B, the function of neuromuscular junctions was evaluated using repetitive nerve stimulation. ASO treatment at postnatal day 25 rescued the decrease in CMAP values due to RNS, as we can see in the mice that were treated with the ASO compared to the vehicle-treated mice, compared to the vehicle-treated mutant mice. Also, motor unit number estimation, which is in short known as MUN, which accesses the number of functional motor units, shows that ASO administration at postnatal day 25 rescued the number of functional motor units close to the wild-type levels in the treated mutants. So reduction of CMAP and MUN in the Bergeron mice are reminiscent of the human symptoms. In the last section of this webinar, we'll feature mouse models and study designs or in vivo pharmacology services uses routinely that can be used for preclinical evaluation of gene therapy. So not only do we offer the largest collection of neurological mutant mouse models, but we also offer a wide variety of preclinical services for many specialized mouse models of rare and orphan disease. As we can see the list on the left side, these are some of our famous neuromouse models for rare and orphan diseases. And the yellow ones, and the ones which are in these yellow boxes are the ones we frequently work with. We can also create new mouse models for rare disease at our Rare and Orphan Disease Center. JAX in vivo services offer a set of well-defined, reliable, robust, and reproducible in-life and post-life assays to capture translationally relevant aspects of gene therapy. We'll start with the featured mouse models and study designs that we have used for amyotrophic lateral sclerosis ALS studies. So for ALS, there are many impaired molecular pathways and many genetic variations, as well as environmental exposure that contribute to the pathology in ALS. The figure on the left summarizes the multiple pathogenic pathways that can affect the key cellular functions of motor neurons, and that includes RNA and protein metabolism, and neuromuscular junction structure and function, cytotoxicity, and inflammatory responses. The graph on the right shows ALS gene discovery since the early 90s. About 10% of ALS cases are transmitted in families almost always as a dominant trait. The first genetic mutation found to cause ALS reported in 1993 affected the gene SOD1. The cumulative numbers of known ALS genes have increased rapidly. The size of each circle here reflects the proportion of all familiar ALS cases associated with that gene. So as we can see, almost like 20% of the familial genes comes from SOD1 mutation. And then we have 45% of familial mutations that come from mutations in C9 or 72. So here is the summary of 5 most characterized mouse models for ALS using JAK's in vivo pharmacology services to evaluate therapeutics. And these models are kind of grouped based on what pathway is affected. So for SOD1, it's the superoxide dismutase mutations that is the characteristics of this mutant model. Then we have TDP43 inclusion models, and then we have mouse models which shows the problems in C9 ORF72 mutation. So to learn more about these models, please check our webinar, In Vivo Drug Discovery Platforms for Amyotrophic Lateral Sclerosis. So here is a sample study design for an efficacy study using the SOD1 G93 ALS mouse model. Mice enrolled prior to the onset, and we take baseline from measurements at 6 to 7 weeks of age for rotarod, CMAP, and RNS stimulation. We can also monitor serum biomarkers as well. And there is we can do multiple dosing routes are possible for these mice. We can measure onset of progression of disease by electromyography and rotarod at various time points. Post-study tissue analysis is available. We can study neuroinflammation using spinal cord tissue, and gliosis can be studied using microglia and astrocytic markers. So autosomal recessive, so the next disease we are going to focus on is Friedreich ataxia. Friedreich ataxia is an autosomal recessive disease. It causes progressive nervous system damage, which leads to difficulty in walking, loss of sensation in the arms and legs, difficulty speaking, cardiomyopathy, and all of these symptoms worsening over time. Currently, there is no cure for this disease condition. Our varied range of treatments are used to treat comorbidities, including medications and physical therapy. Molecular mechanisms that is involved in this disease condition is low protection protein that is important for mitochondrial function. And what happens is there is presence of GA repeats in intron 1 of this gene. The number of these repeats occur between 5 to 30 times in normal individuals. But in case of mutations, this repeats goes from almost 62 to 1,000 times, with more repeats correlated to more severe disease condition. Again, because of these repeats, it causes a reduction of information of frataxin protein, which ultimately leads to mitochondrial dysfunction. So here is the summary of 3 most characterized mouse models of Friedreich ataxia used in JAK's in vivo pharmacology services to evaluate therapeutics. And one of them is the cardiac specific model, which shows cardiomyopathy, body weight loss, reduce activity in the heart. We also have neuronal-specific model where we see neurological decline, progressive decline in motor movements, increase in sporadic and uncontrolled movement, which we can monitor through open field tests. And we also have a mouse model in which we have increased GA repeats, where we can see the mutation in GA repeats expansion, and we can see the severity of the disease correlates to the size of the expansion of these GA repeats. So a sample example study design for Friedreich ataxia model for the neurospecific model, we can measure prophylactic. We can do prophylactic and therapeutic dosing for these mice. We know the exact time points where we can start the prophylactic and therapeutic dosing. Again, multiple dosing routes are possible in these mice. In life readouts, we can do body weight, neuroscore, and we can do it daily as well as weekly. We can also measure. We can also do rotor rod and open field tests to check for ataxia and nerve conduction velocities. Post study readouts are also available where we can measure histology for gliosis and we can do neuron counts. We can also do protein analysis, gene expression, and also vector biodistribution for AAV gene therapies. So the last featured model is a Rett syndrome model. Is a genetic disorder where 99% of the cases is caused by de novo mutation in the X-linked methyl CpG binding protein, which is MECP2. The figure on this left side shows the functions of MECP2 in the brain cells, where it's involved in all kind of transcriptional activation, repression, chromatin loop formation. So all these activities are affected because of the mutation in MECP2, and that leads to the symptoms, the neurological symptoms we see in this disease condition. Since the mutations are X-linked, there is a female gender bias for this disease. Males that have the X-linked mutation usually die in infancy. Currently, there is no treatment for Rett disease, Rett syndrome, but there are treatments for some of the symptoms. Again, physical occupation and speech therapy can be employed. Nutritional support and behavioral intervention can be done to mitigate some of the symptoms for this syndrome. So here is a summary of 2 most characterized mouse models of Friedreich ataxia used in JAK's in vivo pharmacology services to evaluate therapeutics. Hemi males, which are very often used for this in vivo studies, have a survival of 90 days, and they show all kind of abnormal gait, hypoactivity, and hyaline clasping phenotype. We have het males which have a prolonged phenotypic expression that we say highlight clasping and some other phenotypes at like 3 months of age. So for example study design that is used in the hemi males, we can do prophylactic dosing strategies. We can also measure disease onset and progression via daily body weight. We can also look for survival in these mice. We can look for whole body plethysmography to measure the apnea, and electroconvulsive therapy to measure seizure susceptibility, which is a reliable outcome for the disease condition in these mice. Also, multiple dosing routes are at different time points are also available, where we can do prophylactic drug administration at P0 and P1 stage with ICV injections in these hemi males. So to summarize the key concept overview today, gene therapy has come a long way regarding safety and efficacy since it started 6 decades ago. We have shared examples of the case studies using clinically relevant mouse models, which show a biological response to gene therapy products similar to the expected response in patients. Jack's in-depth expertise on neurobiology and in vivo models provides several of the preclinical requirements for the FDA, which includes feasibility, safety, biodistribution, and efficacy. I would like to acknowledge Dr. Kat Lutz and Dr. Lauren Bogdanik and Dr. Maximo Lupresa at JAX. We are very lucky to have these researchers with massive experience developing and optimizing mouse models for neurological disorders to test therapeutics and inform preclinical and clinical trials. I will also like to thank Dr. Michael Trulio, Senior Product Manager, Dr. Dolores Garcia Arocena, Scientific Engagement Manager, and my colleague at TIS, Dr. Corrigan Smothers, for helping me in the development of this content.